Hello, my name is Maitreyee Potdar, and I am a current senior at Las Lomas High School. And from July to August 2023, when I was a junior, I did an internship in Manhattan, Kansas for the Mechanical and Nuclear Engineering Department at Kansas State University. I learned a lot from my advisors there, who are listed on this slide, and would like to present what I learned as well as my findings through this presentation, which is titled 2D Nanomaterial for Inhibition of Bacterial Growth. So first, I'm going to give a brief background on nanotechnology, covering types of nanomaterials, their production, and common applications. Then I'll dive into my research on 2D MoS2 and E.Coli, including the creation of MoS2 nanoflakes, their characterization with microscopy, contact angle measurements, and cell culture studies. So first, we have some basic information about nanomaterials. What are nanomaterials and 2D materials? So nanomaterials refer to materials with dimensions on the nanoscale, which ranges from 1 to 100 nanometers. These materials can exhibit unique properties compared to their bulkier counterparts due to their small size and high surface area. 2D materials are specific class of nanomaterials that consist of especially atomically thin layers. So, for example, the nanomaterials that I mostly dealt with would be at about the size of these nanotube electrodes, which, as we can see, are much, much smaller than the size of an ant. Next, we have what are the dimensional classifications of nanomaterials and how does it affect their properties? So, nanomaterials are classified based on their dimensional structure. 0D nanomaterials have all dimensions at they are represented by quantum dots and just nanoparticles, basically, as can be seen here. One-dimensional nanomaterials are materials such as nanowires, nanotubes, nanofibers, nanopillars, as you can see here. I worked with 2D materials, which are represented by nanosheets, nanoplates, nanocoatings, basically like these layers, this seen here. And there are also 3D nanomaterials, which have all three dimensions larger than 100 nanometers, such as nanoprisms, nanoflowers, as we can see here. And 2D nanomaterials possess sorry, here we go. What are the properties of 2D nanomaterials and how they're different from bulk materials? So 2D nanomaterials possess unique properties such as high anisotropy, large surface area, mechanical strength, and plasmonic behavior. Their atomically thin nature gives them exceptional electrical, thermal, and mechanical characteristics, which are ideal for electronics, energy storage, and also nanomedicine. So, how to make nanomaterials? There are two main categories in which you can use to make nanomaterials. There is bottom up and top down. So, there are fundamental approaches in nanofabrication and synthesis which describe the control and size reduction of the process. So, bottom up strategies involve the assembly of smaller components such as atoms or molecules, which create larger structures. And this approach allows for precise control over the final product and can help lead to novel properties due to the atomic or molecular arrangement. In contrast, top-down strategies involve the reduction of larger structures, such as bulk materials, into smaller ones, allowing for miniaturization of features. So an example I received was that top-down would be like taking a bigger I don't know, like a piece of fruit or something, and then like cutting it into very thin layers. Like you're reducing the bigger material versus bottom up would be as if like you planted a seed into the ground and it slowly grows bigger and bigger into a plant, which is like adding the different components which create the bulk material at the end. So now I'm going to go over my specific research, one facet of it, which was making MoS2 nanoflakes through exfoliation. So one specific method that you can use to make a nanoflake or nanomaterial is called top-down micromechanical exfoliation. So micromechanical exfoliation, or also known as the Scotch tape method, is a top-down technique for isolating thin layers from bulk materials by applying and peeling off adhesive tape. This method famously extracted single-layer graphene from graphite and allows for iterative thinning of materials. While it provides a simple way to obtain nanoscale materials, it has limitations in scalability and precision. So these would be the steps, if just for reference, but it's basically you would prepare the piece, attach material like MoS2 onto the like silicon wafer, for example, and then press the tape, slowly peel, and keep repeating to get thinner and thinner layers as needed, and then you would observe. So I can go over just the quick process. In the first step, you can see me breaking the wafer into the small pieces. Then we are placing the MoS2 flake onto the wafer for stability. Later, then you place the tape on top and remove. You can place it on the copper plate, which is used to observe it under the optical microscope, which I will show later. And this is the microscope that was used for observing these MoS2 flakes. You can see the coarse and fine adjustment knobs where the lens of the microscope was, and the slide on which the flakes were secured, which was placed on top of that copper plate. And through this optical microscope, which was connected to a computer, we were able to closely observe these MoS2 flakes. So this computer had a program installed on it, which allows the lens images of the microscope to be depicted on the screen, which you can see an MoS2 flake depicted there. So these were the results I got by observing these thin flakes under the optical microscope under 5 times exfoliation, so peeling the tape 5 times, 10 times, and then 15 times. And then this was again on a different instance. We have 5 times, 10 times, and 15 times. And again, so we can just see the pieces, the layers getting thinner and thinner because through repetitive peeling of the tape, you get thinner and thinner nanomaterials. So next, I'm going to be talking about the facet of my research that explored how to use a scanning electron microscope to observe these exfoliated MoS2 flakes. So how to use an SEM and what is an SEM? So scanning electron microscopy, or SEM, is a technique that uses a focused beam of electrons to examine the surface of solid objects. When the electrons hit the surface, they create signals that help show the object's shape, composition, and structure. SEM produces detailed, high-resolution images by collecting information from specific areas, allowing for an in-depth study of the material. So these are just the quick steps of how to use a scanning electron microscope. Sorry. Basically, you would prepare the sample by attaching the wafer to the aluminum stub within the microscope. You would hold vent and then replace the blank sample with an MoS2 wafer. You can then press evacuate and wait for the green light to turn on. Then you would adjust the beam to make sure it's on your sample and adjust the zoom settings and capture an image. So here is a photo of what our SEM looked like. You can just see the vacuum chamber here, the electron chamber on top, and the monitor, which is depicting the electron imaging. These were the controls used to zoom in, zoom out, and increase focus and clarity. And this monitor shows the inside of the SEM, and here are the stage controls, which were used to tilt the wafer and sample inside the SEM. So these were just the photos of the procedure, attaching the wafer to the stub, turning on evacuate, um, putting the sample within, and these were the adjustment knobs just to get these images displayed here on the monitors. So these would be the results of observing the wafers using the SDM with 5 times exfoliation and then 10 times exfoliation. And here we have 10 times exfoliation. We can see how the layers appear to be thinner and slightly more see-through. And we can also see how more detailed the images that the SCM provides versus the earlier shown optical microscope. I'm going to cover another facet of my research, which was using a goniometer to measure contact angle of MoS2. So what is contact angle? So when water is deposited on a solid surface, its behavior is determined by the intermolecular forces between the liquid and the solid, which is quantified by the contact angle. So a contact angle over 90 degrees indicates limiting wetting, which also it's called hydrophobic. It means that the material is hydrophobic, while below 90 degrees suggests effective wetting, meaning the material is hydrophilic. If the droplet fully spreads or if it remains spherical, it is termed complete wetting or superhydrophobic. So what is the utility of a contact angle? Contact angle measurement not only describes wettability, but also provides insights into the adhesion properties of coatings, which are crucial for both decorative and protection applications. Strong adhesion typically correlates with optimal wetting, and techniques like plasma treatment and the use of different adhesion layers can enhance surface wettability. So, what is the significance of contact angle measurements? One use, for example, of a contact angle can be in the agricultural industry. One might take a sample of soil and test the contact of water in regards to that soil in order to find the right soil that has the balance of both hydrophilic properties, so water would be absorbed enough to water the plants, and also hydrophobic properties, so the water could last longer to water the plants for longer. So this would be the process of taking a contact angle measurement. Basically, as an overview, would prepare the sample surface by cleaning it, deposit a 200 microliter droplet of liquid using a micropipette and capture a high resolution image. Then you would analyze the image with a specific software and identify the contact angle, the intersection between the droplet, the surface, and the air. And then using the less than 90, which means hydrophobic, greater than 90, which means hydrophobic, less than 90, which means hydrophilic, you would analyze your image. So here are some results that I got when we did this in the lab. So we can see here where the contact angle was taken. Basically, we used the program to draw these lines manually, and then the program would tell us what the angle was. So this first image, since it was less than 90, it's hydrophilic. So is the second image. These images are also hydrophilic, and we can also see it tells us more statistics up here. And this is also a hydrophilic image, and we finally get one hydrophobic image because it is 0.49 above 90 degrees. And then another facet of my presentation is why should people grow bacteria on 2D materials? And in my experiment, we used MoS2 and E.Coli. So why these choices specifically? So for E.Coli, choosing the right biological, oh, sorry, we're going to explain why even grow them first. So certain bacteria prefer specific surfaces for colonization and biofilm formation due to their surface properties. Researchers use 2D materials to study how bacteria interact with these surfaces compared to traditional ones. Growing bacteria on 2D materials helps modify these surface properties. So the nanoscale features of 2D materials change characteristics like roughness and charge. allowing researchers to study bacteria's role in enhancing these changes for applications like antimicrobial coatings and medical devices. So nanomaterials are vital in surface engineering. Researchers use bacteria on 2D nanomaterials to understand how nanoscale features affect bacterial attachment and biofilm formation, leading to advanced surface modifications. So now, why specifically E. coli and MoS2? So choosing the right biological matter is essential when you want to study surface modification by bacteria. And viruses and E. coli was selected due to its lower cost, ease of growth, and well-documented research use. So unlike harmful bacteria, E. coli is generally safer and more widely used as a model organism. And in regards to MoS2, or molybdenum disulfide, it was chosen as the substrate due to its unique electronic, chemical, and mechanical properties. Unlike graphene, MoS2's layered structure and semiconducting nature make it ideal for studying how E. coli would interact and modify on these nanomaterial surfaces. So how is E. coli grown and kept alive? Basically, an overview would be that you have to prepare a growth medium like LB agar or broth, sterilize via autoclaving and add any supplementals and antibiotics if needed. Then you would use a sterile loop to pick up an E. coli colony and place it within the growth medium. You would then use a sterile loop to pick up an E. coli colony from a previous culture and streak it onto a fresh agar plate or liquid medium. Then you would take the plate and incubate it upside down to ensure there's no condensation that transfers onto the plate at about 37 degrees Celsius. And then once the culture grows, you would transfer a small amount to a fresh media and continue the growth. You can add inducers like IPTG to activate or suppress genes if necessary, and then you would just keep monitoring and store cultures for a longer time in glycerol stocks or something for kept at about negative 80 degrees Celsius. So this was the process of making the broth in which the E. coli would grow in. So first, you grab the powder, the LB powder, and you dissolve it in deionized water. You would cap the container, cover it with foil, and then you can see here the autoclaving machine, which you would autoclave for about 121 degrees Celsius for about an hour, making sure to cover it with foil. And then you can cool and store the broth at 4 degrees Celsius for up to a month for use. And how would you know if when you add the E. coli to your broth, if you are seeing any growth? So basically, growth appears as colonies on agar or turbidity in broth. Okay, so on this slide, we'll be looking at how to view these MoS2 E. coli wafers within SCM. This slide's mainly describing the SCM process, but you can view these wafers under an optical microscope as well. So in SCM, you would just rinse off the bacterial cells with a buffer to remove the fixative off this sample. Then you would dehydrate using increasing ethanol concentrations to dehydrate the cell, mount the sample onto the SCM holders, and apply a conductive layer of gold to prevent charging. And then just for the imaging, you would adjust the settings and capture your images. So on this slide, we'll just be looking at the wafers observed under an optical microscope. So in these first two images, you can just see how it's the wafer with the broth. So there's not growth to be seen because there's no E. coli. It's just the wafer. Versus this is 1 day of culture. You can see it's already starting to be covered. And then after 3 days, it's basically completely covered, which we can see again on this slide. The E. coli is basically just covered the whole wafer. And then just to observe the specific E. coli by itself, taken from literature, we can see at 10 micrometers the E. coli, and then closer image at 1 micrometer. So throughout, what were the insights gained throughout this internship and this experience? So First, I had received training on the preparation of the broth solution for growth and culture of E. coli on a variety of surface modified silicon wafers. In addition, in prior to even my arrival at the lab, I had completed a wide range of biosafety and biosecurity courses that helped address multiple regulations and standards and guidelines to ensure I was following proper procedure when I was in the lab. And of course, I contribute to the preparation, dry release transfer, characterization via optical microscopy and SCM, molybdenum disulfide, and graphene layer nanomaterials on silicon wafers for assessing their biofilm inhibition characteristics. And I didn't just here were some images of me at the lab. So this is during the contact angle measurements here as well. This is my ID. This is the SCM, and during the application of the E. coli to the MoS2 wafers. Um, and I didn't just stay in the lab during this trip. I had also visited many of the interesting and amazing nearby places, such as Aggieville, which is really close to campus, that has a lot of cafes and restaurants, which I ate at here, and also a really good donut shop that I enjoyed. And also the architecture of KSU as well is just really amazing. Like this is Hale Library, which I thought just looked great. We also went to the stadium, which was really big. And they have parks nearby as well, which we visited. It had like a little concert playing when we went as well. And of course, we walked in the Konza Prairie Trails, which there aren't many prairie trails that you can walk through nowadays. So this was a really interesting experience. Um, so yeah, thanks for listening. And I would just have some acknowledgments to the professors that allowed me access to their materials and for the image sources that I got my images from. Thank you.